Thank you very much. This is a wonderful opportunity to present what we're doing in Eurofins Discovery. Let me advance to the next slide. Let me try that. There we go. I'll use the bottom cursor. Okay, apologies. So the title of our talk is Harnessing the Power of Translational Cell Models to Provide Solutions to Complex Biological Questions. I'm the Senior Scientific Director of Translational Cell Models, Dr. Eric Schwiebert. Dr. Alistair King, Head of Biology at Eurofins Discovery and the Translational Biology Center of Excellence, is going to present the first two introductory slides, and then I'm going to present some more detailed slides on translational cell models. So Alistair, please introduce yourself and present the next two slides.
Yes, thank you, Eric, and thank you to the organizers for the opportunity to present here. We're very honored and very grateful to be able to do so. So it's my pleasure to give you the background here as to what we will be talking about and how the translational cell models unit that Dr. Schwiebert will be presenting fits into the broader portfolio of translational assays that we offer that really encompass key parts of drug discovery. So as you can see from the slide that is currently showing on the screen, there are a number of different services that can be offered that can facilitate drug discovery from a number of different aspects. So starting on the far left-hand side, and these all constitute different aspects of translational biology or phenotypic assays. So on the left-hand side, we have biomarker services, and this is the ability to modulate cellular systems with modeling a number of different aspects of biology and measuring biomarker changes as a result of exposure of those models to test agents. This might, for example, be treating PBMCs, human peripheral blood mononuclear cells, with test agents and measuring the effect on cytokine secretion. At the top, above the translational biology icon there is the Biomap platform. And the Biomap platform is a very elegant mixed co-culture system that actually employs a number of different models of different aspects of tissue biology and disease to provide a more fuller picture of how a test agent works. At the bottom, excuse me, at the bottom is the OncoPanel platform, which is a cellular in vitro phenotypic platform that very specifically looks at agents that are relevant to oncology drug discovery. And in this respect, it's a very useful platform for looking at agents that show direct anti-cancer activity. Now, there may be the question that can one look at immuno-oncology agents where the microenvironment is evaluated? There are models that can satisfy that need in the Biomap platform above. Now, obviously, the key part of the presentation today is the translational cell models unit, and that provides yet another aspect to the translational biology portfolio by providing models that really capitalize on a number of things. And Dr. Schwiebert will go into a bit more detail about what that is and the value of employing those models. But from a very high level perspective, those models really make good use of human primary cell types, and in particular diseased versus normal tissue. So really getting to the physiological underpinning of these kinds of in vitro models that are used and very important aspects of drug discovery. Dr. Schwiebert will also talk a little bit about aspects of the translational cell models platform, and not only where that interfaces with the other cellular phenotypic platforms that I've just briefly discussed, but also other aspects of drug discovery. And that includes things like looking at ion channels, looking at ADME toxicology, and looking at other aspects of drug discovery that Eurofins can cater to. So if you could go to the next slide, please, Eric. So just a very brief overview here in terms of the four components. I'd mentioned them very briefly. The Biomap platform obviously gives a broader evaluation of the biological consequences of a test agent. So this gives more of a global view of what a test agent is doing. This can encompass things like looking at mechanism of action, especially for test agents that have been identified from phenotypic screening, where the mechanism may not be clear, right the way through to things like toxicity signature analysis. So this is a platform that really can get to predicting with great accuracy and translational reliability a wide variety of properties of a test agent that really mimic and reflect what is seen in the clinic. As I mentioned, the OncoPanel services platform really is beneficial to look at direct anti-cancer agents. So these would be typical oncology drugs that are being developed to inhibit the growth, proliferation, or viability of human tumor cells. The biomarker services platform is a lot more open-ended, and in essence, it is a very flexible and highly customizable platform. And simply put, if one can set up a cellular model, and it might, for example, be one of the OncoPanel cell lines, one of the human tumor cell lines, it may be one of the biomap models that we have, it may be one of the translational cell models that we have, or it may be a specific cell type. If one can set that up and measure specific biomarkers, that is what the biomarker services platform can do. As such, it can provide a lot of information around mechanistic activity of test agents in non-engineered endogenous cell lines. So, for example, if one has a receptor tyrosine kinase inhibitor, one can look at phosphorylation of the receptor. Excuse me. Can look at phosphorylation of the receptor as a direct readout for the mechanistic activity of that test agent. And then last, but by no means least at all, the translational cell models, which Dr. Schwiebert will be presenting shortly, really capitalizes on those more physiological aspects of both human primary tissues, primary cell types, and the ability to compare normal and diseased tissues. So collectively, all four of these platforms provide great assistance to various aspects of drug discovery across a range of therapeutic areas, and really help to, as the title of the slide says, ease that journey from concept to clinic. So typically going from a hit or leads that have been characterized perhaps in biochemical assays, looking at them within the context of these cellular phenotypic assays to really understand what those drugs are doing, and ultimately to candidate selection and really profiling that candidate so as much is known about it as possible before taking it into the clinic. Ultimately, of course, this is an essential part of drug discovery, and with the models that are available, and especially models like the translational cell models, really help improve the chances of success of those candidates when they do go into the clinic. So it's my very great pleasure to hand over now to Dr. Eric Schwiebert, who will continue with the rest of the presentation. Thank you very much.
Thank you, Alistair. That's a wonderful introduction. And it's great to be with you. It's an honor to be with you. Thank you for the opportunity. I'm Dr. Eric Schwiebert. I'm a cell biology, cell physiology, cell pathophysiology expert by training. And what we have as translational cell models as scientific foci are shown on this slide. So in the context of Da Vinci's Vitruvian Man drawing you see on the slide, we have cells from a bunch of different tissues, lung and airways, liver, kidney, vasculature, reproductive tissues, skin tissues, and even sensory tissues, and beginning work in neuronal and in neuronal support cells. And the emphasis that I want to mention here, whoops, okay, we're starting to do that. Okay, so the emphasis that I want to have here, the foci are primary human cells derived from tissue. We're very experienced at this. It also lends to doing isolation of cells from diseased tissue versus normal tissue. I'm going to show you several examples of those. We study these cells in both 3D biogels and in other 3D culture formats, as well as in traditional 2D bioassays where the cells are grown as a cell monolayer. Our diseased human cell models are underpinned by genotypic, phenotypic, and pathological analysis. So you'll see with polycystic kidney disease, and I'll mention that we know the genotypes of the cells that derive from the patient tissue. And we're also very good at customized collaborative projects for clients. So if we need to add particular assay readouts or biomarker readouts, as Alistair mentioned, we have the capabilities ourselves or with our other units that we collaborate with to be able to add in different measurements, assay measurements, experimental measurements into the assays we perform on both diseased and normal human tissues. We also have already identified synergies with some of the other platforms and units, as Alistair has mentioned, and I'm going to touch upon some actual case study examples and models where we're working with other units within Eurofins Discovery. So where can TCM provide value for different types of companies, different types of marketplaces? Well, obviously, biopharmaceutical and biotechnology companies focused on tissue-specific human diseases is one place where we're a very good fit. And we can perform these customized collaborative projects for clients with basically any human cell type, almost any human cell type that they ask for. There are many artificial intelligence or AI drug discovery firms out there that are doing drug discovery against protein structures and are using IBM Watson or different types of AI algorithms or tools to be able to identify new drugs or repurpose existing drugs for new human disease indications and targets. And oftentimes those units have computer experts, but they don't have wet labs. And so we're there as the wet lab to perform experiments for them to see whether the output of the artificial intelligence experiment has actually found drugs that are going to work in that particular human disease indication. There are academic drug discovery cores that do drug discovery themselves, as well as internal units within biopharma and biotech that do their own drug discovery experiments, but they need secondary validation experiments or ADME-tox support or other types of validations or confirmation assays that the drugs that they've nominated as hit to lead and lead candidates truly are viable and can move forward. Companies that cannot use live animals in research are also a good marketplace for us because they need to come to us for the primary human cell models to be able to do those bioassays and experiments. Companies that are interested in sensory testing or in taste testing or in food testing. I'm going to show you some examples of how we've been using human taste cells to look at the effects of different food ingredients or bitter tasting medicines, for example, finding bitter blockers that might block the bitter taste of certain bitter medicines or bitter food ingredients. Skincare and anti-aging longevity companies is another marketplace that we're looking into with cell senescence assays, as well as different types of skin cell assays with different types of primary human skin cells is something that we've done in the past in projects, and we can do again. And then also we're beginning to look at reproductive cell types, human reproductive cell types for both women's health and men's health. These are also cells that we can procure and design and perform assays around. So several different examples of marketplaces and types of companies where TCM can provide value to their projects and to their programs. This is a simplified diagram of a drug discovery critical path or a workflow of experimental steps where one first starts with a cell-based assay or a target or a pathway, and then one screens at least 100,000 compounds in that cell-based assay against a particular target. And a couple of things that we want to talk about in this critical path as we screen through compounds and we identify the hit the lead and lead compounds that come from the screen, which is usually 1% of the compounds that are screened, or about 1,000 compounds in this case. We actually suggest that the client adopt a relevant human cell model to be used to screen on for the entire critical path. That's not always decided by the client, but that's something that we actually suggest and consider, because we actually think the human cells are the key ingredient in a lot of the different bioassays and experiments that we do. Now, in an event, a human cell model that does not exist that that a client or a project wants to screen on, we can create one. So we can create new primary human cell cultures from tissue. We can immortalize those cells to make an immortal human cell line if the project needs one. And so just consider that, again, in both of those cases, the human cell is the key ingredient in our view in doing this. And the final value proposition is the lead optimization, prioritization, and progression. So once you've screened 100,000 compounds, and once you have 100 to 1,000 that you want to examine, this is where we come in and as a unit in Eurofins Discovery, and other parts of Eurofins Discovery come in to try to do these secondary validation assays. We bring our chemistry expertise in here to look at the structure-activity relationship tables of the hit-to-lead chemical classes that are found out of a screen. And we do lead compound profiling in so-called secondary validation assays, where we're doing these experiments on primary human cell models of relevance. If they're diseased, or if they're normal, or if they're both, we do this. And oftentimes, once we identify a hit-to-lead class that really looks good in our hands, the client will do iterative medicinal chemistry. Additional medicinal chemistry analogs will be synthesized, and then those analogs, those medicinal chemistry analogs, those modified small molecules, will be tested again in the system. So there's an iterative cyclical type of secondary validation assays that we can do in this case. And so this just gives you an example in the context of the drug discovery critical path or flow diagram of experimental assays, how we can add value. Let me give you some actual case studies of how we do this integrative human cell and tissue physiology underpinned by diseased or normal human cells. I want to take you through some different types of offerings we have off the shelf and some example data that come from those experiments. So this shows our polycystic kidney disease platform, and it also shows some comparisons of primary human cells versus immortal human cells. So this is a piece of a polycystic kidney that has become enlarged and has become fully cystic. This is an individual dissected single PKD cyst from that tissue, and you can see it's still very round and it's filled with fluid, and that is the bona fide translucent fluid-filled cyst that you actually see from remnant human diseased polycystic kidneys. And so what we do is we isolate a primary culture from each individual cyst from each kidney. And oftentimes there can be as many as 50 individual dissected cysts that we can dissect out of an individual enlarged PKD kidney. So we get as many as 50 different primary cultures from these enlarged kidneys. Each one is grown up in its own culture vessel, and then each culture is tested in a 3D biogel to see how cystogenic the culture is in our 3D biogel system. And you can see an example of that. This is a high content image of the entire well of a 384 well plate. And then we do high content imaging, disease scanning through the biogel to reconstruct the entire biogel and to see every cyst in the well and in every biogel. Primary human cells are virtually exclusively used for these assays because they are established from tissue and they are closer to the in vivo condition. There is a finite lifetime for them in culture, however. They don't grow forever. They're not immortal, albeit TCM has some cell culture tricks to make them last longer and to be passaged for a longer number of passage lifetimes. Now, one can introduce immortalization genetic material into these cells to trick the cells into thinking that they're immortal. Cell immortalization has its benefits, especially if the tissues are quite small and the primary cells that you derive from those tissues are quite limited. That is certainly the case in eye tissues and in taste bud tissues that we also study in primary and immortal cell culture. But the immortal cells have the potential to be expanded into larger quantities and to be placed into defined panels for expanded assay offerings. An example of that is OncoPanel, where Eurofins Discovery has over 300 immortal human cancer cell lines that can be used for anti-cancer agent profiling. The process of cell immortalization, however, can also change the cells that one is working with. So they are no longer like the primary cells from which they are derived. So whenever we immortalize cells, we compare them back to the primary cells from which they were derived to make sure that they haven't changed dramatically. So that just gives you a comparison of primary human cells and their utilities and their limitations, and immortal cells and their positives as well as their limitations. Let me show you some of the data from some of the industry standard drugs that we have run in these assays. This is 3D PKD profiling data, and these are the assay metrics that we measure at the end of a 13-day cystogenesis assay, where the cells are seeded into the 3D biogels. They form cysts over time. The cysts either get larger, or if the drug is effective, it causes the cells the cyst to shrink or even to disappear. And you can see that the drug in red right here shows a cytostatic growth arrest of the cells in the nanomolar range here. So it's showing a lovely result here. This is the 3D cell titer glow data showing viable cells in the well. This is the cyst number that's shown here, where we do the AI algorithm tool and the Z-scanning up through the biogel and the reconstruction, so we can count the number of cysts in each well of the biogel. So that you see the number of cysts has gone down with the red drug. With the blue drug, it has gone down, but it's gone all the way to 0. We get a little bit concerned if the 3D cell titer glow values go all the way to 0, because that suggests it might be overtly cytotoxic into the system. So we can also measure LDH leakage into the media, so we can see the cell viability versus the LDH leakage and discern whether it's a cytostatic growth arresting drug or if it's an overtly cytotoxic drug. You can see in some of these other conditions, we have a drug that's not quite as potent, but is also showing some nice inhibition of cystogenesis, and two drugs that either have been tried to be tested in clinical trials or are approved for PKD show a partial inhibition in the micromolar range. So if one takes this boxed set of concentrations at 100 nanomolar and wants to compare the 100 nanomolar potency of these different drugs and wants to see it in typical images of the cyst imaging that we do in high content imaging, this is shown here. And so the total cyst area in each well is shown in this plot on the left for the 100 nanomolar concentration of each drug. And the typical cyst image is shown here on the right. And you can see that in the DMSO vehicle control, there are cysts present. In these two drugs where it wasn't quite as effective in the nanomolar range, you still see cysts. And in the other cases where the drugs are showing some effect, trend towards significance or actual significance, you can see that there are few cysts, or in the case of this drug, what is happening is that the cysts, the fluid-filled cysts, are actually shrinking back into spheroids. So you can see a lot of darker circles here. These are actually spheroids now instead of cysts. So these are just some examples with some industry standard drugs of how we do these assays for these projects. Now, let me show you some examples of what we do with human taste cells. These are human taste cells that are actually isolated from taste bud tissue and grown in primary culture. We can also immortalize these cells and make immortalized versions of these cells. But one study that's going on right now is that the goal of this study is that the best hits from human cell-based medium throughput screening on these human taste cells are being subjected to safety, validation, and mechanism of action based experiments by multiple different units within Eurofins Discovery in our St. Charles, Missouri location. The client wants all 7 hit compounds to be tested for solubility and tier 1 ADME, which are toxicology assays, to make sure that none of these compounds have any safety liabilities or any difficulty being worked with because of solubility constraints. And actually, the client also wants to verify that the compound truly has the molecular structure that we think it does, coming from the library and the vendor. So we're also going to do LC mass spec and NMR on these small molecules. Eurofins ion channels is going to do a cube-based experiment on a particular family of ion channels to see whether any of these bitter blockers inhibit these particular ion channels. So it's an ion channel assay where each different ion channel is expressed in heterologous cells, and they're going to do an automated whole cell patch clamp experiment to see if any of the hit compounds inhibit this particular family of ion channels. And then we at Eurofins Translational Cell Models, who did this medium throughput screen for the client, is going to do an ATP secretion assay, because we suspect that some of these bitter blockers may be ATP release channel inhibitors. And so we want to do some experiments to discern that. And we're also going to do some primary human cell safety experiments on both the HT-Beck cells, but also oral keratinocytes, upper airway epithelial cells, and esophageal epithelial cells to make sure none of these bitter blockers are harmful to those different specialty human cell types. Let me show you how we found some of these bitter blockers. So the ATP secretion assays, the stimulus is the bitter medicine that we're screening against. So this happens to be a bitter antiviral medicine where we did a dose response with the real-time ATP secretion assays, and you see with increasing concentrations of this antiviral drug, you see a greater and greater real-time increase in ATP secretion, which is highest at the 1 millimolar concentration for this antiviral drug, and it goes up in a dose-dependent manner. There are also some different kinetics to the ATP secretion. There's a rapid phase and a slower phase over the course of 30 minutes, which is stimulated by this antiviral. And then in the presence of this antiviral drug as the stimulus, we then screen small molecules one at a time to see if they would block the stimulation by this bitter medicine. And indeed, this compound on the right, 6-methylflavone, showed a dose-dependent inhibition of the TAF stimulation of ATP secretion in this HT-Beck system. So the important thing is, does this bitter blocker work in the hands of other assays with other collaborators, and does it work in the human mouth? And the answer is yes. So several different bitter blocker candidates from the screen were tested in the hands of another laboratory on HEK293 cells expressing specific bitter taste receptors. And so TAS2R39 turned out to be the bitter receptor that was stimulated by this antiviral drug and that was antagonized by 6-methylflavone, the bitter blocker. And you can see the result here. There's a complete block in the response to TAF, and there's a complete block in the real-time cell calcium signal in the HT-Beck cells. There are other bitter blockers that show partial inhibition, as you can see here in the coded and blinded set, but this one showed the complete block. And so given that, our positive results and this laboratory's positive results, we did testing in human subjects with a sip and spit assay. And basically what that is the antiviral drug is given to the subjects and they hold it in their mouth for 30 seconds, and then they spit it out and they tell the investigator how bitter it was, the perceived bitterness scale. And so this bitter medicine was bitter to both of these subjects. And then in the presence of the bitter blocker, we see almost a complete block. This is the first time that this investigator saw a complete block with a single bitter blocker small molecule. And it was repeated twice in 2 different subjects. And actually, he didn't believe it. So he repeated it 3 more times in each subject. And that's not shown. That's not shown on the slide, but it repeated 5 times in these 2 subjects. So this is an example of an exciting example about how human cell-based drug discovery actually works, because you can do a couple of steps in vitro cell-based assays and then test it in a human subject and show efficacy in vivo. And so we were very excited about that result. With the same HT-Beck cells, we don't just do bitter taste physiology. We also can do sweet and salty-based physiology. And this is an example of what we're doing for a project where we're actually using antibodies to the sweet taste receptors, and we are fluorescence activated cell sorting and enriching the population for sweet responsive cells. And so the first part of the study is to do that, is to biobank some sweet responsive cells that are sorted from the fluorescence activated cell sorter. Then we're doing some buffer optimization to try to understand the best assay buffer to use with these sweet responsive cells. And then we're doing cell calcium assays, ATP secretion assays, and cyclic AMP assays in response to stimulation by a bunch of different sweet tasting molecules that may be flavor ingredients for different foods or beverages. And after those cell calcium assays, ATP secretion assays, and cyclic AMP assays, where we try to ascertain which sweet taste molecules are stimulating those different signal transduction pathways, we have some other studies in mind where we're going to look at sweet taste signal transduction. And then ADME Toxin Biomarker Services is going to help us measure some released biomarkers from the taste cells that may not just be ATP, but they may be other types of neuropeptides, neurotransmitters, and other types of molecules that are released by the taste cells in addition to ATP, which is the main neurotransmitter. Now, if this works, I can show you which is the most exciting part of the presentation, and that is a video of cells happening in cells actually working in real time. So I'm going to hit the play button here, if it will, and you're going to see the screen go black, and then the minute the injection happens, you're going to see the cells light up. Boom, the injection happens, the cells light up. And so most of the cells in this window where we're doing this real-time cell calcium movie are sweet responsive cells that are being stimulated by 1 millimolar saccharin, which is an artificial sweetener. Now, it's a possibility that once you get into the millimolar range, that saccharin can also stimulate bitter receptors. So we can't rule that out. But most of the cells in the image are responding, and you can see that when as soon as the injection of saccharin goes into the well, you can see the cells respond. And we can measure individual cells in these calcium movies, or we can measure the fluorescence from the entire culture, which we're measuring here. And on the right, now that I've shown you the cell movie a few times, this is the actual dynamic read of the same culture, where you can see the injection here. The saccharin stimulates the fluorescence, which is the cell calcium fluorescence. This is a flow 8 AM dye. And then the signal decays over time, as you saw in the movie. And in the presence of lactazole, which is a sweet taste receptor antagonist, that blunts the majority of the signal. That's shown by the dashed line here. So you can see that our taste cells are useful for sweet taste signaling as well as bitter taste signaling. And we also can do salt-based signaling. So let me go to the next slide so it doesn't keep playing the movie. So we are also doing studies in skin cells, with anti-itch or anti-inflammation, or even skin care assays with skin cells, either dermal fibroblasts or skin epithelial cells, also called keratinocytes, where in the first phases, Ion Channel Services is doing hex cell-based assays with a particular type of calcium entry channel to see whether therapeutic candidates affect the function of those channels. So they'll be doing an automated patch clamp electrophysiology assay. At the same time, we're going to use the biomap to try to understand how the therapeutic candidates inhibit anti-inflammatory or immune biomarkers in several different monoculture or co-culture primary human cell models in biomap. The results of those two studies, either in terms of effects on the calcium entry channels or effects on anti-inflammatory or anti-immune biomarkers, are going to inform us in translational cell models how to do our real-time cell calcium imaging assays in human skin cells and in neuronal cells, and also some biomarker services-based assays looking at inflammatory assays in the human skin cells. So we will identify some secreted inflammatory mediators here, and we'll measure them again but in human skin cell monocultures and co-cultures. So this is an example of different units within Eurofins Discovery St. Charles working together to serve the needs of a particular project. We can also do human cell cytotoxicity or human cell safety assays, depending on how you look at it, with a bunch of different human cell types. I showed the Vitruvian Man drawing and the cell types that we can grow from the different tissues. That can certainly be mimicked in a 384 well plate, where one can take a therapeutic candidate and test it on kidney cells, liver cells, excuse me, skin cells and lung cells, to see whether it has any cytotoxicity on those cells at increasing concentrations, or whether it's safe, whether all those concentrations don't harm the cells at all. So that's a very simple 2D assay that we can do at the right, or we can grow some of these epithelial cell types on permeable filter supports, measure the transepithelial resistance, add the compound, and see if there's been any harm to the cell monolayer integrity of those cells using an electrical based assay. And these assays we think can be useful and might reduce the number of live animals used in toxicology testing. If we can test, hit the lead compounds earlier using primary human skin cells from different tissues, perhaps we can find harmful compounds and remove them from further consideration and progression. These are some examples of actual data you can see from the human kidney cell safety experiments. You can see the concentrations at 30 micromolar here, also shown in the dose response curve. You can see that some of these drugs actually showed a significant effect on cell viability at the 30 micromolar concentration. And some of these compounds at even lower concentrations show an overt cytotoxic effect on the kidney cells. And this is a human cortical kidney epithelial cell line, and this is a mixed medullary cell line from the kidney, showing that there were similar responses in both those cases to some of these industry standard drugs. The one in yellow or in gold is a known anti-cancer drug that is quite toxic, not only to normal cells, but also to disease cells, called Velcade. And there's some other examples here where you see partial or full cytotoxicity effects. There's one drug in here, Vertex 809, that didn't show any effect on cytotoxicity and was there as our negative control. We also are starting to get into doing customized co-culture or triculture models for different types of projects. This is a triculture model of 3 different cell types. I can't disclose what they are, but they all come together to mimic bone marrow tissue in this particular case. So we're starting with optimizing, the triculture from cells from 1 donor. We want to solidify that with cells from 4 other donors. We then probably will progress to disease donors versus normal donors using this triculture system. And biomarker analysis and lead therapeutic testing will occur thereafter once we've solidified both the diseased and the normal model for this particular project. And in a related type of approach, we can grow these same cell types on a permeable filter support and begin to think about epithelial permeability testing in monolayers or co-culture systems. Finally, in the last few slides, let me show you what we're doing with primary human mixed cancer cell cultures, as well as cancer-associated fibroblasts, known as stromal cells, in a mixed cancer cell phenotype that mimic the tumor microenvironment. So us, together with OncoPanel and Biomarker Services, are scripting out these projects where we want to test agents that block the interaction between epithelioid cancer cells and stromal fibroblasts. That's definitely a kind of a new area in anti-cancer research. We also want to look at the stromal fibroblasts to see whether we can't begin to develop some cell senescence assays, because cell senescence of cancer-associated fibroblasts, it seems to be a hot area right now. We can use existing cell lines from OncoPanel as well as our own primary mixed cell cultures to create these 3D spheroid model systems, which I'm going to show you in a subsequent slide. We also think that cancer-associated fibroblasts might help in 3D spheroid formation and the reproducibility of that type of assay system. And so we're continuing to develop this. And so this is the mixed cell culture that you can see here, freshly isolated and growing out to confluence. And these are the cancer-associated fibroblasts. And you can see these are very much epithelioid and mixed, whereas these cancer-associated fibroblasts are starting to show a fibroblastic appearance and starting to get, hyperproliferative and can be passaged for many passages. This shows you what we've been doing with our primary human mixed cancer cell models. This is showing the spheroid formation in the middle of the well in only 20 hours. And in 48 hours, it's completely formed here. You can see here across several different wells in the plate, you can see a rather uniform organization of one cancer spheroid per well. And what we would like to do as we continue to develop this, and this is enhanced actually by the presence of cancer-associated fibroblasts in this particular system, is to be able to do high content imaging and to estimate the size of each spheroid in each well across the plate, and have this be an assay that's 5 days long, where we seed the cells in the beginning of a week and then analyze and image the spheroids at the end of a week. It's a lovely complement to OncoPanel, and may enhance the OncoPanel 3D offerings that we currently offer right here. And one can do this in the absence and presence of anti-cancer test agents and be able to do this type of analysis. So let me end here with the last slide. Seems we're pretty much on time here, and just reemphasize the take-home messages. We're very happy to be part of Eurofins Discovery right now. We were acquired almost a year ago. Before that, we were Discovery Biomed. Before we were acquired by Eurofins Discovery, we are now TCM, or Translational Cell Models. Our key core principles are the human cells are the key ingredient, number one. Number two, primary human cells derived from tissue is something that we emphasize and try to do in all of our projects. We do 3D as well as 2D bioassays. And I don't just mean 3D biogels. Those spheroids that I showed you form naturally in the 384 well plates without biogel, actually. So those were 3D spheroids without biogel that formed one to a well. And we hope to be able to continue to optimize that and begin to offer that, greatly enhanced by the inclusion of fibroblasts and an additional cell type. And our diseased human cell models are underpinned by genotypic, phenotypic, and pathological analysis. The 3D PKD cysts that I showed you, we know the genotype of each of our tissues that came from each patient. So we know the genotype of the cells that we're putting into the 3D biogels. We also test each one of those cultures to make sure they're cystogenic. So we've got phenotypic analysis of them as well. And we know some pathology reports from the patients, whether it be from the PKD patients or from the cancer lesion or tumor tissue. We very much like the power of diseased versus normal human cell comparison. From a very early age as a PhD student, and then a postdoc, and then as an academic for a while, and now as an applied scientist with a CRO, I've always felt that the power of diseased versus normal human cell comparison is incredibly powerful. And so I can't emphasize that enough as being something that we can offer here as Eurofins Discovery TCM going forward. And also, let me emphasize this. This is something we're continuing that we did at Discovery Biomed, and we're continuing now, and that is you doing customized collaborative projects with clients. We are happy to design the project with the client's needs and wants in mind. We are happy to go back and forth with the client in designing the project and designing the experiments and designing the assays. So when I mean customized, collaborative, creative, and communicative, I mean it, because we feel like the scientists that we're serving in projects need to be included in what we're doing. And finally, what I'll say is the TCM synergizes really well within the Eurofins Discovery ecosystem. We're already finding standard and customized projects with different units, and we're actually seeking those types of projects that we can serve and that other units within the Eurofins Discovery enterprise can serve. So with that, I will stop. and I will take any questions.